Hello and welcome to the immunology lectures, and we were discussing on the complement pathway. So, we have discussed about the complement overviews in our last class, and if you remember that we told in the last class that the complement pathway can actually be initiated in three different ways. The classical pathway, which is an antibody dependent pathway. Then you have an alternative pathway, which is antibody independent, and you have a lectin pathway, which is also antibody independent. So, it basically does not depend on any antibody antigen interaction. And I also told that all these three pathways, they finally tend to converge at a single point, that is, they try to produce a lot of C3 convertase, which actually cleaves the C3 into C3A and C3B, which are the cleaved products, the cleaved complement products. So, how these pathways they do these things? So, as we told that initially the complement was found or discovered as a system which can complement for the humoral branch of immunity. So, it basically is the effector pathway of the humoral branch of immunity, and it helps to complete the function of the antibodies. So, it is not only that the antibodies binding to antigen will complete the function, it also requires the complementation by some proteins and some factors, and those are the complement proteins. So, the complement proteins can initiate the pathways in three different ways: the classical, the alternative and the lectin, of which only the classical pathway is antibody dependent and the rest two are not. So, let us quickly look into how these three pathways are initiated, and the three pathways they try to break down the different complement proteins and produce the C3 convertase, which finally leads to breakdown of C3 into C3A and C3B. So, starting from here, so if you look in this picture, we have the classical pathway, the alternative pathway and the lectin pathway. Let us consider this portion to be the surface of the cell, that is the cell surface, where this antigen is bound to the antibodies, primarily the IgM, the immunoglobulin M. Now, binding of this IgM, when the IgM binds to the corresponding antigen on the surface of the pathogen or on the surface of the bacterium, that leads to a conformational change. And this conformational change exposes the regions on the Fc region on the antibody. So, on this Fc portion of the antibody, which has binding sites to the complement proteins. Now, to which complement protein? It binds to the complement protein, which the name looks very complicated. If you look, it is the C1q-R2S2. So, basically the C1 complement protein, it comprises of many polypeptide chains, among which this C1q, it comprises of approximately, there are 18 peptide chains which are arranged in a ring like structure like this. So, into 6 ring like structures, and this can bind to the R and the S. So, it basically forms a oligomeric structure in presence of C1q, C1r and C1s. So, basically it is written together as C1q-R2S2. It comprises of the C1q, the C1r and the C1s, and they together forms this complex structure, which is the C1Q R2S2. So, per unit there are 2 C1R and 2 C1S. So, that is why it is R2S2, and this is the C1Q R2S2, and this bar over here usually in a complement activation pathway, this bar usually indicates an active protease or an active protein. So, after these cleavage products, they again try to associate with each other with the target proteins, then they form these active proteins which can cleave further more complement proteins. So, they are indicated by this kind of a bar on the top. So, these are nomenclatures. So, once this C1Q R2S2 can bind to an IgM or the Fc region of the IgM, then it is kind of activated. And since I told that these are all proteases, so they can lead to the cleavage of other complement proteins, that is the target complement proteins which are present in the vicinity. For example, they can break C2 and C4. So, C2 is broken down to C2A and C2B, and C4 is broken down to C4A and C4B. Now, these are the cleavage products, the C2A, 2B, 4A and 4B. So, now it forms, they immediately associates and they form another complement protein which is known as C4B2A. It is the C4B2A. Now, this C4B2A is nothing but a C3 convertase, which means it can convert or break down C3 into C3A and C3B. So, what happens? C1Q R2S2, that is the C1 complement protein. The C1Q R2S2, when it binds to the Fc region of the antibody, after there is an antigen antibody interaction, this Fc region binds to the C1Q, that leads to activation of the C1, leading to cleavage of C2 and C4, and C2 breaks down into 2A, 2B, C4 breaks down into 4A and 4B, and this 4B and the 2A, they associate together to form the complement protein or the C3 convertase, which is also known as the C4B2A. Now, this C4B2A is a C3 convertase, that means it can cleave C3 into 3A and 3B. So, it can cleave C3 into 3A, C3A and C3B. So, now let us look into, for the time being we just stop here on the classical pathway. We go to the lectin pathway. We see how the lectin pathway is initiated and how it is initiated. So, in the lectin pathway, as I told, there are specific lectins like mannose binding lectin, for example MBL, and also there are lectin proteins like phycolin. Now, these can bind to carbohydrates or mannose molecules that are present on the surface of the pathogen, and when they bind to the surface of the pathogen, they can also bind to a class of serine proteases, which are the membrane associated serine proteases like the MASP1 and the MASP2. These are also parts or components of this lectin pathway. So, they can bind to this MASP1 and MASP2, and it forms an oligomeric structure similar to this, and once this binding occurs, they can also convert C2 and C4 into the broken products like C2A, C2B and C4 into C4A and C4B, similar to what we have seen in case of the classical pathway. Now, here again this leads to the formation of C4B2 C4B2A. So, again the C4B and the C2A, they combine. So, 4B and 2A, they combine and they form another intermediate in the complement system, which is known as the C4B2A, and it is also a C3 convertase. Okay. So, now this C3 convertase can also convert C3 into C3A and C3B. So, you see that in both the pathways you have formation of an intermediate which is the C4B2A, which can lead to conversion or breakdown of the C3 into C3B and C3A. Now, C3B is one of the very important complement components. Now, let us see, as I told initially, that the alternative pathway is initiated only when there is C3B present, and you also need the presence of another factor called the factor D. So, you have, let us say, this is the broken product of C3B, that is the C3B, the broken product of C3. Along with that, you have another factor which we call as the factor B, and with the help of a third complement protein also known as factor D, it cleaves the factor B into BB. So, B again is cleaved. Since it is also a complement protein, it is cleaved and forms a large fragment which is called the BB. So, now this BB, after breaking of this factor B, so this B is a broken product of this B, factor B, and this is possible only in presence of factor D and the C3B. So, now this forms another C3 convertase which is known as C3BBB. So, now this is another C3 convertase. Okay. So, all these three C3 convertases are formed at the end of these three pathways: the classical pathway, the alternative pathway, or the lectin pathway. Among these three pathways, if you see, only the classical pathway is an antibody dependent pathway. The rest of the three are antibody independent pathways. Now, if we try to see how these three pathways they converge at a single point. Now, they all try to break down the C3. So, they all try to break down their central role is to break down this C3, and they break it down to C3A and C3B. Again, C3A and C3B. Now, this C3A, the C3B can have, it can work in two ways. One, it can further associate, with this 4B2A intermediate and form C4B2A3B. So, it is just simple addition. So, if you see, it forms another convertase which is also known as C5 convertase. So, now their role is to break down the complement protein C5. Somehow they have to break down the complement protein C5. A second way is they form another C5 convertase by the action of a protein propyridine. So, this propyridine, it can also associate with the C3b and it can form the C3b BB C3b. So, this is also another C5 convertase. So, this is also a C5 convertase. So, at least you have two C5 convertases, here one and here one. So, both of them, both of these C5 convertases, be it the C4b2a3b or so 4b2a, which was originally a C3 convertase, when it associates with another broken C3b, it forms C4b2a3b and this is a C5 convertase. It can break down C5 into 5a and 5b. Similarly, this C3bbb, which is a product from the alternative pathway, can also, in presence of propyridine, it associates with another C3b, breaks down another C3 into 3a and 3b, and that C3b can also associate with 3bbb and forms C3bbb C3b. So, this is also another C5 convertase. Now, their job is to break down C5 into C5a and C5b. So, we come across at least two major components in the complement activation pathway. One is this C3b, which can directly, this C3b can directly coat the pathogen. So, it can coat the pathogen surface and it can assist in the process of opsonization. Or it can further associate with other complement broken products and can form the C5 convertase. This C5 convertase can actually break down C5 into 5a and 5b, and this C5b can then associate with other complement proteins like C6, C7, C8 and C9 to form C5b 6, 7, 8, 9. And what is this C5b 6, 7, 8, 9? It is nothing but the membrane attack complex or the MAC. So, now this membrane attack complex can attack the surface of the pathogen and form this kind of a hole where you have fluids, inrushing fluids can come in and can finally lyse or lead to lysis of the cell. And also you have two other smaller products at the end. One is the C3a and the C5a. So, these C3a and C5a, these together, so 3a and 5a, they together can lead to enhance inflammatory responses. So, they enhance inflammation. If you remember the classes from our inflammation, enhancement of inflammation, you will remember that with C3a and C5a were one of the major components that could work as chemoattractants, and also they can bind to many cell surface complement receptors and lead to degranulation of the granulocytes, leading to increase in the increase in release of the histamine, and that would lead to the increase in the vascular permeability, and then would lead to draw more neutrophils and will increase the inflammatory responses. So, basically C3a and C5a, these two cleaved products of C3 and C5, they enhance inflammation. C5b, which is a cleaved product of C5, initiates the formation of the membrane attack complex. We will discuss in our next class how the membrane attack complex formation actually occurs in presence of C5b and the other complement proteins like 6, 7, 8, 9, and C3b can directly lead to the process of opsonization, that is coating the pathogen or the surface of the pathogen with this C3b, the cleaved product, and leading to phagocytosis. So, this is a very general overview of the complement activation pathways, the classical, the alternative and the lectin. We will go through them one by one once again, and we will try to understand how this complement activation occurs. So, starting again back with the classical pathway, as I repeatedly told, or I am still repeating, that the classical pathway of the complement activation starts only as a result of antigen antibody interaction. So, when there is an antigen antibody interaction, there is a conformational change in the antibody that leads to exposure of the Fc regions which can interact with the complement protein C1, leading to or facilitating an interaction in this region, leading to the Fc region of the antibody to interact with the C1 complement. The C1 complement here is assuming an oligomeric structure, which is basically formation of this structure occurs due to association of the three subunits, the C1q, the R and the S, and they form this kind of an oligomer, of which the C1q itself contains 18 peptides or 18 peptides, which are arranged in a ring like structure like this, and they associate with this R2 and S2 and forms the C1q R2 S2. Now, this C1q R2 S2 can cleave two complement proteins C2 and C4. C2 can be cleaved into 2a and 2b, 4 is cleaved into 4a and 4b. This C4b and 2a, they come together, that means they associate with each other, and once they associate, they forms another intermediate, which is the C4b2a. So, we just retain the C at the beginning for the complement and the broken fractions, the broken fragments 4b2a, and this protein or this intermediate is known as a C3 convertase. The name is given as C3 convertase because it can break down C3 into 3a and 3b. Similarly, coming to the lectin pathway, the lectin pathway, I told it is an antibody independent pathway. So, it primarily starts with the interaction of specialized lectin molecules like phycolin or this MBL, which is a mannose binding lectin. They can interact with this mannose or other oligosaccharide molecules or structures that are present on the surface of the cell of the pathogen, on the surface of the pathogen. So, all these interactions are going on the surface of the pathogen. So, when this MBL or this phycolin, it can bind to the carbohydrates on the surface, that would lead to activation of this kind of serine proteases which are associated with these lectins, the MBL or the phycolin. So, MASP1 or MASP2, MASP1 and MASP2 are the membrane associated serine proteases, remember. Now, when this serine proteases, they are activated, they can also cleave two complement proteins similar to what we have seen in case of the classical pathway. They can also cleave the C2 and the C4, leading to from C2 it forms C2a 2b, from C4 it forms 4a and 4b. Once C2 is broken down to 2a 2b and 4 to 4a 4b, again similar to what we have seen in the classical pathway, there is interaction between the two cleaved products leading to formation of a C3 convertase, which is C4b2A. Now, from the two independent pathways like the classical pathway and the lectin pathway, we get the same intermediate product, that is the 4b2A. 4b2A. So, in this case also we have 4b2A, here also we have 4b2A, and both of these are nothing but C3 convertase. So, C3 convertase which can convert C3 into 3A and 3B. Looking into the alternative pathway. Now, what is the role of the alternative pathway? The alternative pathway is initiated only when C3b is available. How can C3b be available? In two ways. One, it can be available as a product of the classical or the lectin pathways, or there can be spontaneous breakdown of C3 into 3A and 3B, and there can be minimal, very minimum amount of 3B is available. So, the alternative pathway basically is an amplification system. It just amplifies the signal if the cleavage product 3B is available, then only it can amplify the signal. So, when there is 3B available, it can associate with another complement factor or a complement protein, we call it the factor B, and this factor B can also be cleaved. It can be cleaved into B. So, the larger fragment of the cleavage of B is the BB, similar to 3B, 4B, it is the BB. Now, this BB is cleaved in presence of another complement factor, which is the factor D. So, if you see this portion, this 3B associates with a factor B and a factor D, leading to the cleavage of factor B into BB, and now this BB associates with the 3B and forms another C3 convertase, which is C3bBB. So, from the three independent pathways, what we get is 3 C3 convertases, and what they all do is they attack the C3 again, and they try to break down as much C3 possible into 3A and 3B. Now, you see the B products, I have marked them in red, and the A products I have marked them in blue. So, why? Because the 3 B products, either the 3 or the 5, the B products, they are effective in two pathways, that is the lysis and the opsonization. What this 3B can do is, 3B can have two different functions. One, it can again form or associate with 4B2A to form the C4B2A3B. This C4B2A3B is a C5 convertase. It converts C5 into, so it is a convertase, means it converts the target. So, C5 is the target, it converts C5 into 5A and 5B. Similarly, this C3B in presence of another complement protein, which is the propyridine, it can associate with 3BBB and form another C5 convertase, which is known as the C3BBB C3B. So, there are two C5 convertases that are possible and they are formed. One is C4B2A3B and C3BBB C3B. So, these two, what we see in this part, these two are C5 convertases. Like these were C3 convertases, this is a C5 convertases. They are also sometimes known as C3 C5 convertase, because they also still retain the property of C3 convertase, but they are mostly C5 convertase. So, these C5 convertase can then cleave this C5 into 5A and 5B. 5B is a component of the membrane attack complex or the MAC. So, it associates with other complement proteins like C6, C7, C8, C9 and forms the membrane attack complex, and this is this tubular structure that is formed because of association of these complement proteins 5B, 6, 7, 8, 9. So, this is nothing but the membrane attack complex or the MAC, and this MAC is nothing but a tubular structure that is inserted on the cell surface, leading to formation of a hole, and that leads to lysis of the cell. At the same time, the C5, which has been broken into 5A and 5B, the 5B as we see is mainly forming the membrane attack complex, the 5A as well as the C3A, which was also a cleaved product of C3, the 3A and the 5A, these two together, they can be important factors in enhancing inflammation. And finally, 3B, as we have seen here, 3B independently, other than forming the C5 convertase, it can also lead to opsonization. It can go and coat the surface of the pathogen and assist the phagocytic process and lead to opsonization. So, this is a overview of the entire the three different complement pathways, the classical, the alternative and the lectin pathways. I hope you understand this. This is a very general overview how these three pathways are being activated, the cascade of the events that are occurring in the three different pathways, how they are being activated and how they finally converge at the formation of the C3 convertase and breaking down the C3 into 3A and 3B, and finally breaking down of C5 and either mediating inflammation or cell lysis or opsonization. So, we will keep continue discussion about this complement system in our next lecture as well. For today, that's all. So, thank you very much.